Eicosanoid synthesis. Eicosanoids are short-range signaling molecules that act upon nearby tissues to elicit a strong physiological response. They are derived from a 20 to 22 carbon polyunsaturated fatty acid or linoleates, which are obtained by our diet through eating plants. Linoleate is then altered to become arachidonate, which is a precursor to prostaglandins, thromboxanes, and leukotrienes. We find arachidonate attached to the phospholipid membrane, the phospholipase A2 or PLA, then becomes activated by some sort of stimuli which cleaves the arachidonate from the phospholipid into the cytosol. The arachidonate can then take different pathways for the prostaglandin and thromboxane synthesis pathway. The arachidonate gets acted on by the enzyme cyclooxygenase or COX, also known as prostaglandin H2 or PGH2, which is a bifunctional enzyme which uses two diatomic oxygens to convert arachidonate to PGG2. There are two isozymes of COX, COX1 and COX2. COX1 synthesizes prostaglandins that regulate gastrin mucin, while COX2 synthesizes prostaglandins that mediate an immune response such as inflammation, pain, and fever due to illness or cell damage. The COX can be regulated by NSAIDs such as aspirin, which is a suicide inhibitor, and ibuprofen, which causes the COX to become inhibited, leading to a decrease in inflammation, pain, and fever when COX is inhibited, but will disrupt normal function of the COX-1 prostaglandins, which can cause stomach irritation. PGG2 is then acted upon by tissue-specific peroxidases from COX, which will yield PGH2, which can then be converted into different series 2 prostaglandins that act like paracrine hormones, or in the thrombocytes, the PGG2 can be acted upon by thromboxane synthase, which yields a thromboxane A2 or TXA2, a series 2 thromboxane, which is its role is to induce vasoconstriction and platelet aggregation for blood clotting. Prostaglandins and thromboxanes both have a circular 5 or 6 carbon ring, which is why is referred to as the cyclic pathway. The other pathway arachidonate can take is a linear pathway, which leads to leukotriene. Arachidonate gets acted on by lipoxygenase, which attaches on an oxygen to either the fifth or 12th carbon, yielding 12 or 5-HPETE. 5-HPETE can then be converted to leukotriene A4 or LTA4, which can differentiate further depending on the tissue it's being synthesized in, such as leukocytes, the heart, brain, lungs, and the spleen. Leukotrienes are non-cyclic, hence the name of the pathway, with three conjugated double bonds, which mediate inflammation and smooth muscle activity, which can be caused by an irritant.